Hi students, this is Dr. K. Sivaya, Department of Pharmacology, Faculty of Santiram College of Pharmacy. Today we can discuss about digestive system, that is next chapter for you. So coming to the digestive system definition, is a system consists of gastrointestinal tract and accessory organs for to break down the food into small components. So whatever you take in the food, the taken we can call it as ingestion. Then after that it is entered into the mouth. So mastication occurs. As a result, bolus is formed. After that we can swallow it. So the digestion by the two processes, by the mechanical digestion and the chemical digestion. So in the mechanical digestion, through the muscular movement of the digestive tract. So here physically to break down the food into small particles. In chemical digestion, say hydrolysis or other enzymes to break down the food particles. Suppose if you take in carbohydrates, it will break down into monosaccharides, and proteins, it is break down into amino acids. It's mainly depending upon the enzymes involvement. Then if you take in the secretions, so enzymes mainly secreted in the digestive fluid by the digestive tract. Mainly this is for the chemical digestion. After digestion, the absorption takes place. The absorption mainly only small components only absorbed. The main site of absorption is intestine. Then after absorption, undigested material, it is eliminated through the rectum. Then if you take in the digestive system organization, it is mainly gastrointestinal tract and accessory organs or accessory structures. So if you take in gastrointestinal tract, it consists of mouth, oral cavity, oropharynx, esophagus, stomach, small intestine, large intestine, rectum and anus. If you take in accessory structures, teeth, tongue, salivary glands, liver, gallbladder and pancreas. See, these are the structure. So whatever we discussed, GIT tract and accessory organs. Then if you take in the anatomy of GIT, so the common layers present in entire GIT tract. So if you take in the innermost layer is mucosa. That mucosa consists of epithelium, and second layer, that mucosa have the three layers. In that, epithelium, and secondly is lamina propria and muscularis mucosa. So in that epithelium, goblet cells present. That goblet cells secretes the mucus, and lamina propria consists of blood vessels, very few only, and immune cells and nerves present, and muscularis mucosa consists of muscle. Then next, then if you take in the second submucosa layer, in this submucosa layer have the more number of blood vessels, nerves present. Then if you take in after that, in this after submucous layer, we have the muscular layer. In that muscular layer, we have the longitudinal muscles and circular muscles. Because of these, the muscles to contraction after this contraction, the food movement, that movement we can simply call as peristalsis. So these are involuntary muscles. Then after that, if you taken the serosa, so this is a serosa, it consists of epithelium and connective tissue. So this is a lumen. So this is a glands in mucosa, already we said that is goblet. So these are the nerves, how it is involved entire through the GI tract. See how this muscular movement happen. So first one, if you take in the peristalsis, so it is a wave like movement that occurs from the oropharynx to the rectum. So to push the food particles toward the anus. So during this movement, automatically mixing happen. That's why it is a mixing motion in the oral cavity. It is first takes place, and stomach that allows the GI tract to repeatedly break down into smaller particles using mechanical digestion. Segmentation. I will show you the difference between the peristalsis and segmentation after the next slide. The segmentation is in a small intestine contracting and relaxing of independently, not dependently, independently. So it is happening mainly small intestine. Okay, then we can see the next slide, we can observe. See, this is in the from the mouth, if you taken after formation of bolus, here the contraction, here the relaxation, contraction, relaxation, automatically it is happening. It is only like this way, it is automatically happen. This is a peristal. Then if you take a segmentation, it is mainly happen in the intestine. Suppose this is one food material, this is a gray in color. So this food material, it is segmented. Then after that, it reaches into one more segment, then a mix. So this is what is happening next. Then after that, what is happening? So then finally mixed. So this is segmentation, and how it is a process of mechanical as a mixing motion, and how these regulated these activities. So suppose if you take this is mainly it is autonomic nervous system. You know, already we discussed autonomic nervous system. So parasympathetic nerves stimulates the GIT tract, and sympathetic nerves inhibit the GIT tract, and also some hormones also to control the GIT tract. Okay, and reflex mechanisms also to maintain the either stimulation or inhibition. We can see step by step. See, so how the nervous control. See the, if you observe simply, if you saw, if you see in any food material, a smell, a taste, thought of food also, it stimulates the directly central nervous system, then directly to stimulate the local enteric nerve plexus and affect the smooth muscle of gland, and response to change in the contractile or secretory activity, either contraction or secretory. See, if you see in any sweet, then automatically your saliva secreted. If the food material, so you automatically, when it is secreted, automatically contraction also it is happening. So that's why it is immediate. This response is immediate response. Then if you take another response is a long reflex. See how when the food material reached into the either stomach or intestine, the chemoreceptors, osmoreceptors, mechanoreceptors will stimulate it. So these stimulates the central nervous system. Then again the central nervous system to activate the local enteric nerve plexus. Then after that, effectors means these local enteric nerve plexus stimulates the smooth muscles or gland present in the intestine. I understood my point. Here once again I am repeating. So when the food material reached into the gastrointestinal wall. That reflex, it produces some reflexes. It reached to the chemoreceptors, osmoreceptors, mechanoreceptors. So that is in central nervous system. Then after that, the efferent action is stimulates through the nerves. That nerves are enteric means intestine. So intestinal nerves are stimulated, then automatically releases the acetylcholine, then automatically smooth muscles will be contracted. If the acetylcholine reaches into the gland will secrete. So then automatically change in the contractility and secretory activity. This is the how the nervous control of the GI tract. Next, mouth and oral activity. So we have to discuss about the entire detail. So food enters in gastrointestinal, everybody know. So the food breakdown mechanical digestion using mastication. So mastication is a process by mixing of mucus with food material, then to form the bolus. So in mouth we can observe the enzyme is especially amylase that is present in saliva. So that saliva secreted 3 glands present in mouth. We can discuss about that glands. Next slide. Next, the tongue made up of skeletal muscle manipulate the food during mastication. So it also contains the taste buds to detect the taste sensation. So then automatically the lump of bolus is formed. It is entered into the pharynx. Coming about the teeth. So all of you compulsory to draw these images, essential. So adapted for mechanical digestion in the oral cavity. See, we have in 20 deciduous or primary teeth before the age of 6 years old. So then after that 32 permanent secondary teeth we can simply call as. So they are divided into 4 types. So that are incisors, canines, premolars and molars. The formula is 2 1 2 3. Okay. So already given. Next. See, in the mouth we can observe the salivary glands. So 3 pairs of salivary glands. Submandibular glands present below the mandible, sublingual glands present beneath tongue, and parotid gland also present in just near to the pharynx. So here these glands entire to secrete the saliva. The saliva helps to moisten the food during mastication, dissolve the food in forming the bolus, and help cleanse the teeth. So what saliva consists? 99% water. So remaining what is that 0.5%? Amylase enzyme, bicarbonate and pH, bicarbonate and electrolytes. So the, if you observe that pH is 6.5 to 7.5. So these are the diagram is very essential. So this is sublingual gland just below the or beneath the tongue. Then if you taken the submandibular gland just below the mandible. Then if you taken parotid gland, it is backside of the mouth or backside of the next posterior to the tongue and just near to the just near to the pharynx. Okay. So these are the ducts open into mouth. So these are the ducts open into mouth. So this is a structure of a diagram, the salivary glands. Then coming to the next step, stomach. So stomach is a jet-like and pouch-like organ. So designed, so it is stored in 2 to 4 hours. The whatever you take in the food material, it stores 2 to 4 hours. So here the some mechanical and chemical digestion also occur. But in mouth, maximum the mechanical digestion happen. chemical digestion, very only one enzyme, that is amylase, and only carbohydrates only digestion takes place. Okay, coming to the stomach have the two sphincters, cardiac sphincter and a pyloric sphincter. So cardiac sphincter, it is entering of stomach, esophagus to stomach, near to the esophagus to between the esophagus and stomach. The pyloric sphincter, it is present between the stomach and small intestine. So it is divided into four regions: cardiac stomach, fundic stomach, and body stomach, and pyloric stomach. So sometimes few they can write either cardiac stomach or fundic stomach, but we can see what is the difference in the diagram. So in that the lumen have this some folds, we can call as rugae. So it is mainly for the expansion, secretion and digestion and some absorption also. So if you observe, this is the stomach structure, this is cardiac, this is esophagus. So here the whatever the small node here, this is a cardiac sphincter, it is present. This is we can call as this portion we can call as a cardiac stomach, this portion we can call as fundus, and this portion we can call as body, and whatever it is here, this portion we can call as a pylorus. So this is a pyloric sphincter. So that's why I said between the stomach and the intestine have the pyloric sphincter, between the esophagus and stomach we can have the cardiac sphincter. Fine. See here also, whatever we discuss in the structure of the GIT in general structure, here the muscle have the one more layer is in the muscle layer, in the muscular, in the muscle layer we discuss. Actually longitudinal or circular layer will be there, along with here oblique layer also present, oblique layer. See the outer layer is more greater curvature, the inner is lesser curvature present. Okay. So this is we can call as a pyloric antrum. Okay. So this is the structure. So remaining the anatomy of the cardiac structure similar to the whatever we discussed before. Then, so what's and cells present in gastric secretory? Chief cells. So chief cells secretes the pepsinogen, and also one more also is there, chymosin. And parietal cells secretes the hydrochloric acid and intrinsic factor, which helps the absorption of vitamin B12 in the intestine. So these two secreted by parietal cells. And mucous cells secretes the mucus, it is mainly to help to neutralize the HCl and gastric juice. And G cells secretes a hormone called gastrin, which stimulates the parietal cells and overall gastric secretion. So this is gastric cells. So how to say? So suppose these parietal cells secretes these parietal cells, mitochondria also given. See the chief cells secretes the pepsinogen, and parietal cells secretes the HCl. So these HCl is convert the pepsinogen to pepsin. So when the HCl contact with pepsinogen, it is converted into pepsin. Pepsinogen is an inactive form. Now I understood, I think so. So this is how the cells, and we can understand what is the difference between the normal general structure of the GI tract and stomach. So come to the what is the digestion and what absorption takes place in the stomach. So carbohydrates digestion because of gastric amylase. In mouth also secreted amylase, in stomach also secretes the amylase, resulting the carbohydrates converted into disaccharides. It is converted into disaccharides, not into monosaccharides. Only disaccharides are formed. So proteins digestion begins with pepsin. So activation of pepsin by HCl resulting peptides are formed. Only peptides are formed, that is small chains of protein. The lipid digestion begins with the gastric lipases, which can only break down certain lipids such as buffer fat, resulting in fatty acids. So little bit it is happening here. So absorption of in the intestine is limited, where only small and fat soluble substances can be absorbed: water, alcohol, aspirin and certain drugs. So in the stomach, the absorption takes place water, alcohol, aspirin, certain drugs, means which have the lipophilic, they are easily absorbed. Then come to the result of all these mixing, chemical digestion, secretion and absorption is a yellowish paste called chyme. So here in the stomach chyme is formed, which will be passed on to the small intestine. So regulation of gastric secretion, see the regulation by the parasympathetic nerves and gastric hormones, and how it is happen through the three phases: cephalic phase, gastric phase and intestinal phase. Suppose if we take in cephalic phase, see it is mainly involvement of sight, smell and taste. Food cause stimulation of vagus, making the brain automatically acid secretion, then automatically parietal cell secretes the HCl. The vagus to stimulate the more stimulation of parietal cells and G cells. G cells secretes the gastric hormone. The gastric hormone increases the HCl production. So then if you take in gastric phase, so gastric phase is a distension of stomach, means enlargement of stomach. When the food material enter into the gastrin, automatically the vagus nerve stimulated, the vagus nerve stimulates the secretions, then automatically the gastric juice formed, and whatever we discussed earlier how it is digested, it is happen. So gastric secretion after if the HCl, if it is more production, it is happen, the gastric secretion inhibited when pH of gastric juice falls below to 2.5. So how it is fall down? Because of sympathetic activity, sympathetic system is activated. So when the pH reach into 2.5, sympathetic system activated and inhibit the gastric secretion. So intestinal phase. So intestinal phases involve the acetic chyme passing into the small intestine, which secretes intestinal gastrin hormone to inhibit the gastric activates. See here we are discussing about in the intestinal phase, when the food material enter into the intestine, the stomach will be empty. When the stomach will be empty, the sympathetic system activated and inhibit the gastric secretion in the stomach. Once I am repeating the point, when the food material enter into the intestine, the sympathetic system activated, inhibit the gastric secretions in the stomach. That is the reflex. And one more important point is when the chyme enter into the duodenum, it secretes the secretin. That secretin hormones that inhibit the gastric acid secretion. So that's the involvement. So this is the entire intestinal phase. So here we can observe the different stimulatory events and inhibitory events. Cephalic phase, sight and stimulation. So stimulates the cerebral cortex through the vagus nerve, stimulate the stomach. And gastric phase, so reflexes, whatever the food material enter here, that sensory, it reach to vagus reflexes and enter into the medulla oblongata, then through the vagus nerve, and again stomach will be stimulated and secretes the HCl and gastric hormone. And the food cells, Food cells when touch into G cells, the C cells releases the gastrin hormone. The gastrin hormone released to increase the more HCl and more digestion and more chyme it is formed. Then intestine phase. When the food enter into the small intestine, that is duodenum. So when it is entering duodenum, the stomach will be empty. That it inhibit the release of gastrin hormone or HCl through the sympathetic stimulation. And one more important point is when the food material, that is chyme, formed in the stomach enter into the intestine, the secretin hormone released. This secretin hormone inhibit the gastrin hormone. So these are inhibitory events. So let's see, when you observe the when the patient loss of appetite, depression, loss of appetite, so there is no capability of HCl so due to because of loss of stimulatory impulses to parasympathetic stimulation. So when the patient undergoes depression, or when the patient undergoes depression, automatically sympathetic system is some is activated, cerebral cortex, so there is no activation, then automatically there is no stimulation of a parasympathetic. When the parasympathetic not stimulated, there is no release of HCl and G cell stimulation. So here also, that's whatever we discussed already. So this is the, thank you for watching. Next I can give the questions regarding for this. For every student we have to submit the answers for the questions, whatever I given questions compulsory. Those are submitting the answers, we can give the present, otherwise we can give the absent. Thank you.